The cell is the smallest unit of life, and within the cell is the cytoplasm. So you have the phospholipid bilayer, or the plasma membrane, which is functioning as that boundary to separate the intracellular components compared to the extracellular components. And what we're going to discuss are the different organelles that are found within the cytoplasm. So let's first start off with the mitochondria. So everyone knows the mitochondria as the powerhouse of the cell. And when we say it's the powerhouse of the cell, what we're referring to is ATP synthesis. And ATP synthesis uses a couple of different processes in order to produce ATP. So a couple of different ones include the electron transport chain as well as oxidative phosphorylation. And in this process, we're using oxygen as our final electron acceptor in order to produce ATP. So mitochondria, that's what I have shown here within this cell. And then now let's move on to ribosomes. So ribosomes are the sites for translation. Translation is whenever we are converting our messenger RNA into protein. So these ribosomes, they contain a large subunit as well as the small subunit. So as this mRNA transcript gets fed into this ribosome, from there we'll then end up producing our protein. So these ribosomes, they're found studded here at different regions. So they stud the rough ER, so that's this component here in red, and then they're also found here within the cytoplasm. So ribosomes, this is the site for translation. They contain the large as well as the small subunit, so they're found on the rough ER as well as in the cytoplasm. So let's look at the rough ER. I've already said the rough ER, it's studded with these different ribosomes here, and it's the site for translation, where we're producing proteins. So as we synthesize these different proteins, from there we need to package them. And the organelle that's responsible, so the Golgi complex, also known as the Golgi apparatus, this is what's known as our UPS. So it's the UPS for packaging these different proteins, and then from there they can get transported to their designated spot within the cell. So now let's move on to the smooth ER. So the smooth ER, this is this component over here. We've already discussed the rough ER, and then coming off of the rough ER is the smooth ER. So that's what's shown here in blue. So the smooth ER, it has a couple of different functions here. One is to detox. So it helps to detoxify substances that enter into the cell. Another one is lipid synthesis. So when I talk about lipid synthesis, what I'm referring to are some of the different types of lipids. So we can synthesize phospholipids, we can synthesize cholesterol, and once we have that cholesterol, that can then be used to synthesize steroids. Steroids including testosterone as well as estrogen. So now let's talk about some of the different organelles that are going to bud off of the smooth ER as well as the Golgi complex. So the first one that we'll talk about, I've already mentioned detox. Well, what comes off of the smooth ER is what's known as peroxisomes. So you can see this peroxisome here budding off of the smooth ER, and these peroxisomes, they contain a couple of different enzymes. And these enzymes include oxidase enzymes as well as catalase. So let's look at a specific example here for the use of peroxisomes. The liver functions to detox some of the substances that are in the blood, and when we consume alcohol, that alcohol or ethanol has to get converted. And so it gets converted and it becomes more oxidized, so it's more water soluble and it can get eliminated from the body. So how does the liver go about doing this? Well, the primary reaction here, it occurs within the cytosol. So 90% of the alcohol that we consume, we start here with ethanol. From there, it gets converted to acetaldehyde. From acetaldehyde, it then gets converted into acetate. Okay, so like I said, this is occurring here in the cytosol. This part right here where we're converting it into acetate, that occurs here in the mitochondria. Okay, but let's look at the role that the peroxisomes as well as the smooth ER play in detoxing the liver. So I've already mentioned the enzyme here that's going to be found within the peroxisomes. It's known as catalase. So catalase, what it does, so we're still using this reaction here to convert. However, we're also using hydrogen peroxide, and as a result, we get the production of water. So peroxisomes are doing this role here, converting it, and then we also have within the smooth ER, cytochrome P450. So this is a particular enzyme found here within the ER, and during this reaction, what the result is, we get the production of reactive oxygen species. And so these reactive oxygen species, they have damaging effects. So they produce what's known as these free radicals, and these free radicals, they can damage DNA, they can damage proteins. So we have to take care of these reactive oxygen species. And this is the part where oxidase enzymes come into play. So oxidase enzymes, right, once again, oxidase enzymes, they're found here within the peroxisomes. So the different enzymes involved here in converting ethanol into acetaldehyde, the enzyme responsible for this is what's known as alcohol dehydrogenase. So the enzyme that's involved to convert acetaldehyde to acetate is what's known as aldehyde dehydrogenase. So aldehyde dehydrogenase. So we've talked about peroxisomes. Now let's move on to something that buds off of the Golgi. This is what's known as lysosomes. So lysosomes, these are larger organelles that are going to bud off of the Golgi, and they're found highly populated in what's known as macrophages. So macrophages, these are what's known as phagocytes. Phagocytes, they engulf things, they break them down. So for instance, if there's a bacterial infection, these macrophages will engulf that bacteria and break the protein down into the different components. So within these lysosomes, there's a particular enzyme, which is known as acid hydrolase. So we're using this in acidic environment within this vesicle, this lysosome, acid hydrolase, in order to break the components down. And the other thing I wanted to mention is what's known as the endomembrane system. So the endomembrane system is the connection between a couple of different organelles. So you have this nuclear envelope, which is surrounding the nucleus. If you can see here, this part in purple, it's converging here with this part in red, right? So all these different organelles, the nuclear envelope, the rough ER, as well as the smooth ER, including the Golgi, all of these organelles here are going to be connected in that endomembrane system. And then what buds off of them, we've already talked about the lysosomes budding off of the Golgi, as well as the peroxisomes here budding off of the smooth ER. So that's the endomembrane system. So now let's move on to the cytoskeleton. So the cytoskeleton has a primary role as far as cell structure, as well as motility of the cell. And there's three main components of the cytoskeleton. You've got microfilaments, intermediate filaments, as well as microtubules. So the first one here, microfilaments, what are they primarily composed of? Microfilaments are composed of a protein which is known as actin. So actin has a major role in a couple of different parts. So when we talk about motility, we can refer to muscle cells as well as a process during mitosis. So within muscle cells, actin is a component as well as myosin where we get the formation of our cross-bridge cycle. So during this cross-bridge cycle, the muscle is going to shorten and lengthen. So that's for the motility. For mitosis, during mitosis, we get the production of two identical diploid cells. And in order to cleave them apart, that actin forms a cleavage furrow, and then those cells are going to break apart. So that's for the motility aspect of it. Actin also plays a role in the structural component. So as far as for structure, known as microvilli. So microvilli are these projections that are found on what's called the apical surface of the cell, or the top part of the cell. They function to increase the surface area. The site where these microvilli are found prominently is within the intestinal cells because we have to absorb these different nutrients. So now let's move on to the intermediate filaments. So intermediate filaments, a couple of the different proteins that include keratin. So keratin is one which is found in our hair, skin, and nails. And another protein is the lamins. So these lamins, they provide a structural support for the nucleus. So I'll write intermediate filaments, structural support. So now let's move on to our microtubules. So with microtubules, they are composed of a protein which is known as tubulin. And tubulin is the quaternary structure that's composed of two main components. So you have the alpha as well as the beta subunit. And so these alpha and beta subunits, they form tubulin. And then these microtubules contain what's known as kinesin proteins. So kinesins are these motor proteins that are going to be responsible for transporting substances within the cell. So these kinesins will travel along these microtubules with a vesicle that contains one of the components of the cell. Building up these microtubules, tubulin is the component of microtubules, and microtubules can be used to build what's known as our centrioles. That's what makes up flagella as well as cilia. So with flagella, the only flagellated cell is the sperm cell. So sperm cell. And then for cilia, this is found within the trachea. So within the trachea, we have the cilia in order to trap these substances. So these goblet cells within the epithelium of the trachea, they're going to secrete this mucus, trap it within that cilia, and then we can get it out of the trachea. So the last topic to discuss is the centrosomes. So these centrosomes are pairs of centrioles. And centrioles are composed of 9 triplets of microtubules. So what these centrosomes do, they have a vital role in mitosis, which is cell division. So what these centrosomes are going to do, the different microtubules that are coming off of these centrosomes, that's what's going to enable to pull these different sister chromatids apart.